Hello everyone, today it is my honor to present a research about a 3D printed sustainable polylactic acid and polylactic acid beta tricalcium phosphate scaffold grafted with the fibronectin proteins for the improvable regenerations. My introduction starts with the bone defects, which are the second highest demand for surgeries in the world, and the bone resorptions of the alveolar ridge have a significant impact in the oral health and the systemic health. And therefore, in this study, we use the 3D printed PLA and the PLA β-TCP scaffold. And according to our previous study in the 2022, we discovered that PLA and 20% β-TCP was the optimal concentration to be used for the osteogenesis. And the aim of my study is to fabricate a 3D printed bone graft scaffold to obtain a precise fit for the patient-specific personalized bone regenerations, and also to improve the osteogenesis properties by grafting with the fibronectin proteins. And in these slides, we can see the property of the material, starting with the PLA, which was derived from the renewable biomass of the cornstarch, and therefore has the sustainable and the eco-friendly properties. And the β-TCP, on the other hand, has the hydrophilicity and the osteoconductivity. However, they still lack the osteoinductivity, and we try to fulfill these properties by coating with the fibronectin proteins, which were bioactive, and to enhance the bioactivity and the response of the osteoblast-like cells. And these slides illustrate the figure of the detailed sample preparations procedure, starting from the material preparations, 3D printing, and the fibronectin grafting process can be seen in this figure. And in here, these are the printer software and the 3D printed samples material, including the bone graft scaffold for our experiments, which were printed out in our lab. And after preparing the final samples and obtaining the final samples, they were analyzed for the physical and biological properties, starting from the SEMs to the gene analysis by the qPCR. We performed both the physical and the biological analysis were tested in this experiment. And in result, the SEM images show the human bone mimicking patterns, indicated that the material having the β-TCP within the PLA, and after 60 days and 150 days, the degradation rate was significantly increased by the presence of the fibronectin proteins. And in here, both the EDEX and the HBS data illustrated the increased carbon and oxygen due to the presence of the fibronectins in the sample group. And in FDI analysis, absence of the bands around 3,500, demonstrating the absence of the byproduct of the PLA, and a multiple peak between 800 to 1,200 contributed for the β-TCP, which demonstrated the successful incorporations of the β-TCP into the PLA materials. And in here, 3D immunofluorescence assay and the MTD assays also showed the highest proliferation rate was found with the PLA fibronectin group from day 1 and day 5, and which also had the statistically significant with the other sample group. And cell differentiation marker of ALP also in line with the MTD assay, showing that the PLA fibronectin group has the highest cell proliferations activity from day 1 to day 5. And in gene analysis, only the DLX5 and SP7 gene expression can be observed in day 1. Other genes such as the collar genes, the OCN, RANX2, OPG, and the ALB expression can be seen on day 5. And their results also showing that the PLA fibronectin group has the highest upregulation for gene expression and have the osteogenesis properties. And to be concluded, we successfully fabricated the PLA and the PLA β-TCP scaffold with the improved cell proliferations and the cell differentiations. And the 3D printed biomaterial also have the sustainable and the eco-friendly properties. In addition, the osteogenesis property through the early osteoblast differentiations marker of the DLX5 and OCN gene expression pathway can be seen. And these improved findings support the future potential application for the customized bone regeneration. Thank you everyone for your attention, and I hope you enjoy my presentation. And if you have any questions, don't hesitate and please let me know.